Good day to everyone joining us and welcome to today's X Talks webinar. Today's talk is entitled Considerations for Neuroscience Trials with Direct Administration to the CNS. My name is Sonia Hunt and it's my pleasure to be your X Talks moderator for today. Today's webinar will run for approximately 60 minutes. This presentation includes a Q&A session with our speakers. This webinar is designed to be interactive, and webinars work best when you're involved. So please feel free to submit questions and comments for our speakers throughout this presentation by using the questions chat box, and we'll try to attend to your questions during the Q&A session. Now, this chat box is located in the control panel, and that's found on the right-hand side of your screen. If you require assistance, please contact me at any time by sending me a message using that chat panel. At this time, all participants are in listen-only mode. Please note that this event will be recorded and made available to you for future streaming on xtalks.com. At this point, I'd like to thank Medpace, who developed the content for this presentation. Medpace is a scientifically driven, global, full-service clinical contract research organization providing Phase 1 through 4 clinical development services to the biotechnology, pharmaceutical, and medical device industries. Medpace's mission is to accelerate the global development of safe and effective medical therapeutics through its high science and discipline operating approach that leverages regulatory and therapeutic expertise across all major areas. Now it's my pleasure to introduce you to your speakers for today's webinar. And first, I'd like to introduce you to Dr. James Voronov. He's a Vice President of the Medical Department at Medpace. James is a board-certified neurologist and internationally known clinician scientist with over 30 years of neurology and psychiatry clinical development experience. At Medpace, he provides hands-on medical leadership to sponsors and internal study teams engaged in neuroscience clinical research. He has worked in multiple neuroscience therapeutic areas, and James received his BA in biology from Columbia University and his MD and PhD from Emory University School of Medicine. He trained in neurology at the Johns Hopkins Medical School. Now it's my pleasure to introduce you to your second speaker, and he is Dr. Felipe Rodriguez. He is the medical director at Medpace and provides clinical and scientific expertise and hands-on experience within neurosciences programs. Felipe provides ongoing training, particularly focused on the operations group supporting the Medpace team during complex neurology and psychiatry trials. He has over 6 years experience in drug development, from first in human through dose finding and early efficacy to pivotal trials. And our third speaker is Kelsey Carter, clinical trial manager with Medpace. Since joining Medpace in 2014, Kelsey has applied her background in clinical pharmacology, pharmacogenomics as a lead CRA and CTM across many diverse phase 1 to 3 trial designs. Kelsey has significant experience in cultivating a strong rapport with sites and KOLs. As a Medpace CTM, Kelsey's experience in clinical operations has optimized processes and timelines throughout the entirety of her studies. She earned a BS in pharmaceutical science and MS in clinical pharmacology degrees from Ohio State University College of Pharmacy and Medicine. Now, it's my pleasure to pass over the mic and the controls over to James. So James, when you're ready, you may begin.
Okay, so thank you all for attending. It's really a thrill for me to be able to talk about this subject with everyone, because everyone who works with me knows that this is one of the areas that I am most excited about in drug development these days. So next slide. So we're going to talk about this idea of directly targeting delivery of therapeutics to the CNS. And then what I really want to try to do, and our other panelists will do, is drive it down to exactly how we do this, because I think the overriding message that I want to give everyone is that this is something that is an exciting breaking area of research, but it's eminently practical. And actually, the truth is we've been doing it for a long time. So go to the next slide. So let me give you first a perspective about kind of where we've been and where we're going so that you can see this area really in context. Go to the next slide. So as you heard, I've been doing CNS drug development for quite some time, and everyone who works in the area knows how difficult it is. You know, there are loose correlations between the pathology going on in the brain and clinical symptoms in neurology and psychiatry. We know that a lot of different pathologies, especially as we've entered the era of molecular medicine and gene um, therapies, you know, they cause many of the same symptoms and work through final common pathways. And so we generally tended to focus either at the symptomatic side or on trying to find common pathways to treat diseases. Now we're in this genetic link, but of course, many of the diseases we treat still are idiopathic without links to causative agents or pathology. But let me focus on the last one, which is certainly what it seems we spend much of our time as drug developers on, is just that we seem to have a hard time getting drugs to the CNS. And actually, not only to the CNS broadly, crossing the blood-brain barrier after being orally bioavailable as a pill, once-a-day pill, but the area of the CNS that we're interested in. So sometimes we can engage the target that we're interested in, but very often these drugs get through the entire brain. We get off-target pharmacology that makes the drug not tolerable or not really useful therapeutically. So go to the next slide. So, you know, getting drugs into the CNS, yes, it's difficult, but as one of my mentors, Sal Snyder, always liked to point out, nature's been doing this for a long time. We have opiates, THC and the cannabinoids, cocaine, even metabolic products like levodopa. They get into the brain. There's different mechanisms by which they get into the brain, transport, diffusion, but a lot of drugs that we developed early on, kind of just by accident, phenothiazines, penicillin, all turned out to be CNS penetrant without or actually trying very hard to do it. Now, our knowledge of medicinal chemistry and, of course, receptor biology and neuroscience in general spawned our entire industry of pharma. And we've been on this quest most of my career of trying to get small molecules to get into the brain, hit the exact receptor and brain area that we want them to, and work their therapeutic magic. So over on the right there, you can see, you know, the early era of CNS drugs, you know, where all these cocaine bearing drugs, like as you can see on that list, if you can read it, Coca-Cola, but lots of other elixirs, powders, headache cures, morphine, cannabis. These were all drugs that were used in patent medicines before, of course, the establishment of laws and regulations to develop drugs with good evidence of efficacy and safety. Go to the next slide. So it's kind of interesting. I just want to start out with anesthesia because as far as I can tell, the first real synthetic agent that was CNS penetrable was ether. And it actually was, these alchemists were fooling around with alcohol and adding acids and chemicals. And so one of the products that you get is ether. And you don't have to be around ether very long to know that it affects your brain. It wasn't until 1846, of course, that Crawford Long demonstrated that you could use ether for general anesthesia. And over on the right, you see one of these early devices. So this is an ether pot. You light a little alcohol flame underneath, warm the ether up to get the vapor going, and that the tube at the end actually went in the patient's mouth. So they could breathe the ether, become, have a lower level of consciousness, and you can operate on them. So chloroform actually just a couple of years was synthesized early, and it actually generally was easier to use. You could just take chloroform, soak it in a rag, put it over the subject's nose and mouth, and have them just breathe, take it on, take it off, and keep them at a state in which it was useful to do anesthesia. But actually, I didn't realize, go to the next slide, that very soon after this, in 1898, you know, cocaine was known as a drug, but its local anesthetic properties. And it was very shortly after that that August Beer demonstrated that you could administer cocaine intrathecally and cause loss of sensation without loss of consciousness through what is spinal anesthesia. So he actually did 8 patients at first. He did amputation with no pain, with full consciousness. Then actually, he had it administered to himself by his assistant, and he administered it to his assistant. And actually, that was probably the first good medical documentation of the post-LP headache. I have no idea what kind of needles they were using, but I assume they were pretty large gauge needles. And pretty soon it was discovered that morphine, when combined with the cocaine, also had good spinal effects. So you have to control position so that you don't get too much movement of the cocaine and the morphine up into the CNS. But spinal anesthesia, as you can see from this advertisement, became a competitor to chloroform and ether as a method of anesthesia. So back in the early days of anesthesia, direct administration to the CNS was a useful method. Go to the next slide. And of course, this intrathecal delivery, when you think about it, is still part and parcel of not only spinal anesthesia, but 1991, the first battery-operated pump was approved, and there are 3 drugs approved for administration through intrathecal pump: morphine, ziconotide, which is a peptide, and baclofen. But off-label, other opioids are used, fentanyl, et cetera. Clonidine is used, and local anesthetics are used. And so as long as one regulates dose and watches position, this is an extremely useful therapy for patients with intractable pain that is generally in the lower regions of the spinal cord. It's interesting, been at Medpace now for going on 8 years, talking to tons of biotechs. Every once in a while, we talk to a company that's kind of thinking about maybe we should do our drug delivery this way. But as far as I can tell from clinicaltrials.gov and some other sources, it's not really been explored as a method of drug delivery for small molecules or peptides, besides what's already been approved. Go to the next slide. And of course, on chemotherapy, administration is well established. In 1963, Dr. Omaya developed the Omaya reservoir, which really made practical this way of administering intraventricular chemotherapy. And intrathecal therapy, through lumbar puncture, is one of the salvage therapies, if you will, for leptomeningeal carcinomatosis, when a cancer spreads widely throughout the meninges, and you want to get relatively high levels of a cytotoxic agent directly to those tumor cells. An LP and administration is one of the ways. One of the points I wanted to make, and as I talked to these companies, et cetera, is stability is an issue in a Omaya reservoir or one of the implantable pumps. It's up against the body, so it's sitting there at room temperature. And so you need a stable drug at room temperature or being able to, flush. And so it's much more difficult than a drug in a bottle or a pill that you can do standard stability testing on, knowing what the stability of your drug is. And so that's one of the interesting things, that what we have for use this way are all known drugs that are being delivered through novel routes rather than new chemical entities where we have many more regulatory hurdles to overcome. So go to the next slide. So it's interesting when you start looking into the history of this. So as we've been doing trials here, I kind of got interested in kind of what adverse events were known, what happened when they did this. And it turned out that in the 40s and 50s, there seemed to be kind of a rage, a trend of physicians trying like everything that they had available to administer intrathecal. And this is from a paper published in JAMA, the dangers of intrathecal administration. And it's a very nice actual review about how the intrathecal space is frequently used for administration. You can see it, serums, drugs, antibiotics, and anesthetics. And things don't always go well. And you know, what they saw then are things that we've seen since in some of the clinical trials that have gone on. So, they tried convalescent serums, right? In the COVID era, we've been using convalescent serum early on as a treatment. Well, it was thought maybe polio could be treated by using convalescent polio serum. Arsenic and metals were developed as antibiotics and used for other treatments. So those were administered intrathecally. It's kind of interesting, penicillin, it was realized pretty quickly that it had good CNS penetration when you measured in the CSF penicillin concentrations from its systemic administration. But as you may know, early on, penicillin was in very sparse supply, was being reused. And so one of the ways, if someone had a CNS infection and you wanted to give penicillin, one of the ways to conserve the penicillin was actually to administer intrathecally so that you got high CNS levels. And the adverse events of high doses of CNS penicillin were well established during this period. But just to kind of overall, aseptic meningitis is the most common adverse event across all of this. Just a cellular reaction in the meninges, presumably, of cells and protein because of these chemicals that are not normally there. The cells respond, and generally these are not extremely severe unless the compound is particularly toxic. And after the compound goes away, it was established in the 40s and 50s, again established in current studies that have been published, publicly presented, that these things go away over time. Let's go to the next slide. So I just wanted to make a note about small proteins and antibiotics. So, I think that we have adequate evidence that systemically administered antibiotics can reach extracellular CNS targets. And I just put the Biogen Alzheimer's drug from the label on amyloid removal from the CNS that's clearly dose dependent, time dependent. I think that we know of pharmacokinetics and antibodies is that what happens is there's tons of antibodies circulating. There are mechanisms for antibodies to get into the CNS, and then the avidity of the antibody for its target is so high that kind of by mass action, the antibodies do reach their target. There have been antibodies to other targets that are in the CNS. For example, I always think about the NGF studies that were done. Did they get into the CNS? Were there CNS effects? It's kind of hard to know. The studies were all stopped because of other kinds of adverse events that were happening in the periphery. But I and people I respect in the field really don't think that you can target intracellular targets using antibodies. But again, I think that thinking about systemically administered antibodies as a way to get into the CNS is something that has not been explored all that much outside of Alzheimer's disease and amyloid as a target. And just for small peptides, we've had tons of programs and no small peptide for CNS, for systemic administration getting into the CNS has ever been approved. And so it's just not clear that that's a viable method. But of course, direct CNS administration could be a completely different story. It's just something that has not been looked at to very much extent. Next slide. Okay, so now Scott talked about natural crossing the blood-brain barrier, talked about the intrathecal route, and now what we're seeing tremendous explosion in is stereotactic technique. So I just wanted to show, this is on the left is Clark's. It's in a museum somewhere. The first human stereotactic frame. And on the right, you can see, we work at Medpace with ClearPoint. They're working on a lot of these trials, like how far we've come now in a century with, computer robotic guided targeting, frames with MRI, use of gadolinium to show administration of the target, the spread at the target. This is really, I don't want to say it's a mature technology, but even from where we were, you know, when I started my career in drug development and we were doing stereotactic administration, as I'll talk about, our technology now to do this is just so much further advanced than it's been. And so lots of what we did before, you know, they were initial trials, but it is research, and the technology clearly helps us drive what we're able to do in the future. Next slide. So this just sort of brings back memory. You know, this is 2005. You know, it's an absolute, you know, Amgen did stereotactic administration to the caudate of GDNF. There were a couple of trials. This was the last one that was published. And, you know, it failed. And on the left, I have from one of the internet, you know, news sites, a little bit about this controversy. But, you know, it's remarkable. You look at the data, there were only 48 patients in this study. And, you know, Amgen, there was no real evidence for efficacy. There was subjective feelings that it was helping. And the trial, you can see, is 6 months of data. You know, they had 12 months in some of the studies. But I think now we would look at this as an early safety study, and not, especially what we know now about the time course of Parkinson's disease and how long it takes and how many patients. And, you know, and I think the Ionis Roche ASO trial really shows you, if you do a trial in, say, Huntington's disease, which is more rapidly progressive probably in that population than in a standard early patient, Parkinson's patient, you know, how many patients it takes and how long it takes to really see the trajectory of patients over time. And we have, of course, in Alzheimer's disease, a wealth of information now. Next slide. So I want to touch on nusinersen because it is truly was a landmark, and I believe it is what is principally driving so much of what we're seeing now in drug development. And look at all these first, right? First approved treatment for spinal muscular atrophy. First approved drug to correct a genetic splicing defect. First approved nucleic acid therapeutic for a neurologic disease. It's the first disease-modifying drug for a neurodegenerative disease, and the first drug to demonstrate prevention of onset in pre-symptomatic patients. You know, all the things that, again, in my career, we've been looking for. This therapy as an intrathecal ASO actually achieved all of those things. So it's great for us because it was a proof of concept that showed that this can be done in the right disease with the right target and the right therapy. And now we're looking for more conditions where we can be successful. Next slide. It's tough to translate, though. This is actually a very similar slide that I used to show 20 years ago, talking about failed stroke trials. You know, you can see the human brain compared to, on the left these are like corner sections. There's nothing small, you know, there's a cat there, and there's not actually a mouse and a rat. Actually, the mouse is there, a rat's not there. But look at the difference between a mouse brain, where most of our translational work is done, and the human brain. And then on the left, you can see the scale. So when you're thinking about like intrathecal administration and the distribution of drug, what you see in a mouse is, what do we really know about what that means? Now, I don't really have time to talk about it, but we're really seeing a growth in the use of sheep as a brain that is larger, is gyrate, that may be a good way to give us an idea of you know, what distribution is like. Next slide. But we do know that, so I just want to talk about, so the other thing we're using now are infectious agents, right? So it's another use of natural delivery. Rabies is a virus that's adapted to go into the CNS and widely infected. You know, prions are proteins that you can ingest orally, target your CNS. Lentiviruses were studied largely, there were trials of lentiviruses. But now I think everyone's kind of settled on this AAV vectors as ways of gene delivery for non-replicating systems. And both intrathecal and serotaxic routes are being used. And, you know, the trials, when we do this targeted therapy, generally the tolerability has been quite good. And I'm really not going to talk about, systemic administration to get into the CNS, because I think that the way the field has moved is to go intrathecal or stereotactic. Next slide. Now, even in our animal models, and these are just some examples, I could have pulled out dozens and dozens of these animal models showing what happens when you use AAV, intrathecal, intracisternal is used a lot in the animal models. You get expression, but if you look at it with a critical eye as a drug developer, the expression is, let's say, just modest, not widely distributed, maybe even in certain areas, not in other areas. So this may depend on distribution, uptake of virus, expression of the vector, et cetera. And we have zero way of looking at any of this right now in people that we are treating with therapies. Next slide. So one of the like sort of where we are now and where we'll be tomorrow, I think, but it's where we are now. I think everyone's realizing that one of the ways that we can start doing better targeting is think about connections, because we know that these vectors can be transmitted transsynaptically. So if in a disease, there's connections between areas and you can pick an area to deliver your therapy to, and then over time have that therapy distributed to the connecting areas which were also affected by disease. Because we know that all the diseases are not limited to single disease areas, brain areas, that this is going to be a promising area, but not every strain is the same. And so we have companies that are looking at strains, looking at capsids, looking at delivery systems to say, how can we quickly and efficiently get the kind of spread through the brain, at least in these animal models, that we want to achieve in humans. And I just want to say that dose, even in the animal models, is something we don't understand. We're just using particles per mil. That's not standard pharmacokinetics as we know it. And it's really an empirical march towards, can we get effect? And is there a level at which there's toxicity from delivering too much therapy? Next slide. And so, again, we're going to talk next about the practicalities of doing all of this, but I just want to make the point that this is nothing new. We do it every day in medicine. We have well-established techniques. There are clearly things that we need to do better, chronic administration, optimization of dose and distribution. One thing I wanted to mention is that, which I didn't really talk about at all, is that, before there was Amgen doing GDNF, there was embryonic stem cell therapy in the substantia nigra. I think that with CAR-T, with CRISPR, we are entering another era of cellular therapy. And I think that in the coming years, and I know there are companies out there working on this, that we are going to see that as a new delivery system that may be better than ASOs and AAVs if you can get engraftment and non-proliferation, but a steady change and integration into the nervous system that actually fixes what's broken. So let me finish with always my sense of urgency, which is that patients are waiting for these therapies. When we roll these trials out, patients sign up. I have incredible respect for them and their families in terms of wanting to push the science forward and maybe get some benefit from these things that we are still really learning about. So I'm going to turn it over now to Felipe, who I'm very happy. I'm working with him closely. He joined Medpace not too long ago out of academia, but he joined out of being a trialist, doing exactly these kinds of trials at one of the major centers that we've been working with. So let me turn it over to Felipe's perspective from the site point of view.
Thank you, James, for the great introduction and overview of the different modalities. My name is Felipe Rodriguez, and I'm a medical director at Medpace. I've been working on the academic and industry side of clinical trials with direct CNS administration for the last 8 years. And in this talk, I will briefly touch on some important aspects that need to be taken into consideration when implementing these techniques. I will start with general considerations that are common to all the different direct CNS delivery routes, and then follow on to specific considerations about intrathecal, intracerebroventricular, and intraparenchymal delivery of investigational products. A brief note that there are many other delivery methods, but I won't expand on those today. We all know that running a clinical trial is a multifaceted activity by itself, and utilizing these techniques can sometimes add an extra layer of complexity. Intrathecal delivery is very similar to lumbar puncture with an added injection. This is the simplest technique of the three and the one with the most accumulated experience. Several medical specialties are familiar with the procedure, such as neurology, infectious diseases, anesthesiology, and hemato-oncology. However, only anesthesiology and hemato-oncology usually will have done injections into the CSF space, and the others need to be trained on this particular aspect. Intracerebroventricular delivery is exactly the same as the procedure done in clinical practice to insert a ventricular catheter in a reservoir, and neurosurgeons are usually experienced. It is likely the most labor-intensive of the three, involving 2 surgical moments and repeated administrations. Intraparenchymal administration is certainly the most complex and involves neurosurgeons specialized in stereotactic surgery and more differentiated technical resources. Moving on to the general considerations that are applicable to each and every technique. We always try to involve sites experienced in the research, the indication, and the procedure. However, this is not always possible, and over the years we developed robust training materials and plans to capacitate investigators to assure patient safety. It is also important to develop detailed and easy to follow study procedures such as a drug administration plan. Some of these techniques will mimic clinical practice, but not all, and may require specialized hardware and other equipment. It is essential that sites can access the means needed to deliver the intervention. It may be a commonplace, but sites and investigators with good past performance tend to perform better. And there should be a very direct line of communication along the study to help troubleshoot any potential difficulties. We make an effort to establish long-term relationships with these sites and investigators. If good participant selection is important in any clinical trial, here is paramount and should be part of the risk management and mitigation strategies in terms of safety and compliance. Given the additional procedural risks and investment on each individual participant, we work with sites to help them screen patients and participants. And these may involve a pre-screening process, review of the inclusion-exclusion criteria, interpretation of laboratory and imaging tests, and review of comorbidities and concomitant medication. Consenting is usually a detailed process, not only for the IP, but also for the procedure, and should involve an explanation of all different steps and risks. There is an extra effort put into developing all patient-facing materials. Also, sometimes we are talking about pediatric populations and adult populations without capacity. So a study companion or a legal representative need to be consented as well, and are essential to communicate with the patient, sustain study compliance, and early detection of complications. Frequently, these studies are conducted in tertiary centers, and participants will be traveling to the site for a visit that is spread over 2 or 3 days, sometimes more. We empower sites to take responsibility for all organizational aspects associated with traveling and accommodation. In terms of the procedure, each will have its specific needs and risks, but all may be affected by things like COVID-19, where being flexible and following local guidelines is paramount. Post-procedural traveling is sometimes seen as a limiting factor for some of these techniques, and it is our role to guide sponsors and investigators on this aspect. Part of the mitigation is to make sure that sites know how to prevent, identify, and manage complications, and that adequate resources are available, such as a 24/7 helpline, inpatient and imaging facilities, and a theater. A large fraction of the indications using these platforms are rare diseases, and in our experience, these may bring some benefits. Patients are usually followed in tertiary centers or centers of excellence that are used to the research studies, are GCP trained and compliant, have the right capacity and the right facilities. Being clinical centers, they know very well their population, and patients are very used to travel. sometimes domestically, but some other times internationally to the study sites. Most indications will have an active service user organization that can help operationalize study procedures in a participant-friendly and acceptable way, and are a great resource to identify high-performing sites and publicize the study. Finally, due to the unmet needs in this arena, regulatory agencies are very open to discuss and are flexible, being very important to engage with them earlier. Moving into the specific delivery methods. For the purpose of today's presentation, intrathecal delivery refers to lumbar spinal intrathecal delivery, and not the broader concept that relates to the administration into the cerebrospinal fluid in general. The procedure is very similar to the lumbar puncture or spinal tap. And because of that, we always look for sites and researchers with large number of LPs done in the past. Systems need to be in place to evaluate and manage complications, including access to an emergency department, imaging, spinal patches, inpatient facilities. Ancillary services are also needed, such as a pharmacy to store, prepare, and deliver the IP, and labs such as CSF lab to prepare samples. It is important to explain that a research lumbar puncture is a very different experience from what is the popular knowledge of the procedure in an emergency setting, and it's very safe. This is most frequently done under local anesthesia, so participant collaboration is essential. In terms of eligibility, criteria are very flexible. There aren't many contraindications, and usually an experienced operator is successful in most circumstances. Before starting the procedure, there's a minimum set of requirements to guarantee safety, including having a normal coagulation and a normal or unchanged neurological exam, although most of these studies will also have brain imaging, which can help us exclude a space-occupied lesion in this context, but this is not strictly necessary. There are some geographical and cultural differences on how this procedure is done in clinical practice. Some centers will do it with image guidance, others without, some with anesthesia, others without anesthesia. Same thing for the positioning of the participant. For the implementation of these studies, we must emphasize the most important thing is a good balance between standardization and flexibility. There is unclear evidence, if any, about some of the procedures that are historically done to minimize the risks, but certainly using a small bore, a traumatic needle, and isovolumetric injection seem to be protective. Traveling after the procedure is usually fine, unless there's a post-lumbar puncture low pressure syndrome where traveling should be postponed. Intracerebroventricular delivery is when the investigational product is injected directly into the brain ventricles, which are liquid-filled cavities inside the brain and represent normal anatomy. This technique is slightly more complex than intrathecal delivery and involves a broader team, including a neurosurgeon, that inserts the catheter, and also a team that administers the investigational product, but that may be the same or a different team from the surgical team. It also involves more specialized facilities, usually a surgical theater, and neuronavigation devices. In our experience, the most important risk mitigation aspect is the existence of an independent surgical committee that helps site PIs and coordinators with surgical eligibility and planning aspects of the study. Although the surgical procedure mimics the procedure done in clinical practice, there's significant regional and inter-site variability, and a detailed step-by-step surgical procedure manual is needed to limit that variability. It is always done under general anesthesia, so that needs to be taken into account when selecting participants. And it's important to note that almost always involves two surgical moments, one for the insertion and another one for the removal of the catheter and reservoir. Brain imaging is essential to plan the procedure, so participants with contraindications are usually excluded. Infection is an important risk, and the surgical procedure is frequently preceded by an infection prophylaxis protocol that can either be local or a central study protocol. And for obvious reasons, coagulation also needs to be normal for this procedure to be conducted. We always recommend for repeated injections to use a reservoir, and that each injection is isovolumetric, meaning that the volume that is collected is equal to the volume that is injected. After the surgery, the positioning of the catheter needs to be confirmed before the first injection. And usually the first injection happens on day 1 post-op, which is also the same day patients are discharged from the hospital. The actual injection is done by a specialized team and under a strict infection control protocol. As with other methods, the teams need to be aware and know how to identify and treat potential complications. And it's important that the participant lives relatively close to a neurosurgical site. It doesn't need to be the research site, but the site with capacity to identify and treat potential complications. Moving on to my last slide and the most elaborate of the 3 techniques, intraparenchymal delivery, where investigational product is injected directly into the brain matter. This also involves a neurosurgical team, but in this case with experience or willing to be trained in stereotactic brain surgery, and an operating theater with intratheater MRI scans and also stereotactic equipment. Again, it's very important to involve neurosurgeons in the development of the protocol and the study plans, as is the existence of an independent surgical committee to help with eligibility and surgical planning. Because the procedure tends to be very long, up to 8 hours, the anesthetic risk is a limiting factor, as is the actual volume of the target being injected. And these are usually confirmed during the eligibility phase of the study. The procedure itself can have several particularities that are worth mentioning. One of them being the use of convection-enhanced delivery, which is a method of injecting the investigational product that not only makes use of a concentration gradient, but also of a pressure gradient to deliver the investigational product over a larger area of brain matter. Another consideration is that for early stage studies, the regulators usually are happier with a staged unilateral followed by bilateral procedure to evaluate risks first. And also the control arms requirements and specifications depend very much on the geographic location and indication. And finally, we always suggest adding gadolinium to the investigational product to get a real-time understanding of where the drug is being distributed. After the procedure, usually the participants stay in the hospital for 1 to 2 days for observation. And although there are potential procedural complications, these are relatively rare in experienced and well-trained hands. Before handing over to Kelsey, I would just like to emphasize that although these techniques may add some complexity and risks, we have plenty of experience implementing them and making their execution seamless, and we are here to help. Thank you.
Hi, this is Kelsey Carter. Thank you, Felipe, for handing that over to me. So now we will start with the site perspective. So we've gained some insight on the history of CNS administration, and we've looked at modern day applications. And now we're moving to how can we implement at the site level and how to operationalize all of these very complex aspects and achieve positive patient outcomes here. So for our site overview today, we will be looking at aspects of the site matrix, study setup, site engagement, and then startup submissions in this first part. Then we'll go on to operations and then open it up for some Q&A. Next slide. And so for that site matrix, especially now that we know some of the procedures and techniques, you can imagine there are many aspects and components to consider. We have in the center here, we have the core site team and the investigator, of course, but we also have many departments to execute the orchestration of these procedures. So at the top of our circle here, we have regulatory contracts and budgets. Very important aspect of setting up the study, understanding at the site level what are the departments we need involved based on how we're delivering these new products, but also considering our patient population. So we move on to pharmacy clockwise. Of course, as Felipe said, preparations with gadolinium, we could have some pre and post-op medications to consider. So pharmacy will be a key component, as well, of course, as neurology, as far as the department's radiology as well, imaging, especially as we establish baselines for these studies. We also may have a dedicated research nurse or practitioner or staff. So we certainly have a clinical team, but we may have a dedicated research department to make sure that, especially towards that long-term care, if patients will be observed for maybe a day or two after, we'll need that dedicated research staff and that handoff there. There's also anesthesiology and surgery. So depending on the technique of the CNS delivery, we certainly may need surgical and then anesthetic teams to oversee, especially considering lumbar punctures and depending on the standard operating procedure at the site as well. Certainly with laboratory, we also now have to consider the potential that we'll be collecting local or central CSF samples as well. So laboratory coordination, stability of samples, and as well as samples that might need very quick processing. We'll need, again, orchestration and teamwork to ensure that we have everyone in the loop. And then as discussed, there's the post-procedure, and this could go to long-term care. So we could, certainly patients may be inpatient, then go outpatient. Should we consider physical therapy, occupational therapy teams, or even home care to support minimizing those post-procedure, any complications, but just ensuring too that our participants feel supported through, prior, of course, during, and then of course after any procedures. One aspect before we proceed to the next slide I want to mention is, depending on the study setup, you could also have blinded and unblinded site teams. So that could potentially expand this site matrix here of all of our contributing parties. Depending on a study setup, depending on a protocol, you may have, blinding that's necessary. So considering the site blinding plan with these personnel will be key. Next slide. And so moving on to some other aspects now that we know our site matrix and we know the potential departments on the site team. And again, we're keeping that main investigator core team in the middle there. And then if we start at the top, when it comes to facilities, qualification of those facilities, we certainly have we certainly have experienced sites, but things in facilities and capacities can change. So ensuring that all of those departments and equipment, all of those spaces are qualified and have well-developed processes that can be applied throughout. We also have the, going clockwise again, primary and backup personnel, access to study-specific procedures, materials, as well as training. And so really here, understanding that we have our primary staff, but our backup staff as well, making sure all are trained on the study, but also for the study-specific procedures, having that handoff So we certainly may have a primary staff team, but if someone should fall ill or if we need a backup last minute, making sure we have that team and ensuring that any evaluators that are doing complex procedures, that they have all been trained in the same manner. So we'll talk a little bit about study-specific training a little bit later. We want to ensure consistency in the same team, not only for the organization of the study, but also I'm sure participants like to see the same team members and faces where possible. The next item here, we spoke to our raters, trainings, and certifications, but the setup of the visit-specific items. So ensuring that they are simplified as much as possible for the site team. So specific kits or supplies, and ensuring that each visit has a flow sheet, source, and using the potential of a mock run to have that be implemented into process improvements, testing it out first. And so that way you have a guide of what can minimize any burden on patients, caregivers, sites, minimize any rescheduling. There's also a key consideration to provide a preferred order of procedures, and then, like I said, have maybe mock runs improve upon that. But that's always helpful. When it comes to safety event reporting and guidance for all departments, so we certainly know that we all have a team obligation to safety reporting and documentation, but that may mean something different for a radiology department or imaging versus our LP procedure team. So ensuring that all of those key roles are aware of their reporting requirements, and as we all know, that should always flow through the investigator, but just for promptness, having them be aware of what, a checklist of what they need to document and report should anything occur. I also have a consideration for local and central travel services. So we talked about minimizing burden on the participants and caregivers. So ensuring that they have support getting to the clinical site or to an imaging site or surgical site, ensuring that all of those other logistics are minimized for them as much as possible. So certainly recommend to provide this support to the subject caregiver and of course any family as a part of that team for the patient. And then a consideration for setup too is there could be multiple days in department training. So we, our site initiation, our study initiation, and our routine visits at site, we'll do due diligence to make sure all of those team members are aware of current study status, any ways we can improve, and that may require some more time just given how big the team is on the study. And so on one hand, we all understand that everyone's busy and we need to be succinct and mindful of time, but on the other hand, we still want to make sure that all of the departments are attended to, they feel they have current updates, and they feel they can confidently execute the study at the site level. Next slide. And so when it comes to engagement, site engagement, so because of the undertaking of the complexity of these procedures and how large our site matrix might be, it's important to keep everyone motivated and informed. I think the key for planning as well is understanding how processes change. Are there new realities? So for site engagement, ensuring on the left here, strategic investigator meetings and periodic site teleconferences to make sure that the state of the study is understood and that site teams feel informed, just also to ensure that they are thanked, appreciated, and understand their hard work and what it means for the study at the study level. Of course, there's typically periodic newsletters or updates. Again, another way to just ensure that site teams are updated. We understand with time commitments, attending an investigator meeting may not be feasible depending on schedule. So ensuring that there are references that are sent for that quick review. And then we also have manuals, plans, and a consideration for study startup is these are complex studies. So we may have additional committees. We may need to understand the role of that study caregiver in our consent forms, any legal authorized representatives, and also those country and institutional specific regulations. This may result in multiple types of contracts and budgets that, again, set up the study correctly. Next slide. So now let's go a little bit deeper into operations in our final minutes here. Next slide. So high level, I have some considerations for administration of IP management, setting up trainings, any specialty lab considerations, that CSF collection, vendor management, and then of course, ensuring a pathway for recruitment and retention. Next slide. So we spoke on these a bit here, but for study site and setup, the key takeaway here is ensuring that everyone's aware of the many moving parts, using checklist plans, flow charts, and of course, making sure there's support to coordinate any departmental setups and trainings. Next slide. A key for surgical considerations for not only intrathecal, but also stereotaxic delivery. We spoke a bit to that mock run-through, ensuring there's a surgical run-through process, keeping in mind that there are planning committees, which may require time and implementation of any observations from surgical planning committees. Making sure there's access to subject data and also study updates by the surgical teams. We talked about travel arrangements, ensuring there's a window of time. And then post-surgery, ensuring there's a debrief with all the teams. Next slide. So we talked about some of the setup for intrathecal delivery here, making sure the communication pathways are open, providing a manual of not only IP admin, but CSF collection. We may need the additional guidance from neurologists, maybe a dedicated lumbar puncture team, providing kits and materials to simplify the process, but also standardize it. Video demonstrations are also very helpful, especially if the IP administration is complex. And then again, any post-LP training on or post-surgery training on, mitigations moving forward. One key takeaway here too is a protocol specification of any coagulation or platelet considerations and testing prior to that lumbar puncture, which likely is already in a standard operating approach. Next slide. We spoke a bit to specialty testing and CSF collection. So the key here too is we may have other vendors that have that study-specific confluence with the study. So raters and evaluators, making sure they have that kit or that packet to address with patients as a part of orchestrating the study execution and procedures. Next slide. Vendor management. So this is kind of an extension of our site matrix, right? And this is where really the operations team can support making sure site teams have the best approach for some of these procedures by central specialty labs, as well as other study vendors. Next slide. So the last two slides here as we finish up are just related to recruitment strategies. So we'll leave this here for your reference, but it's really just to speak to a patient-centric approach with their caregivers, making sure depending on the therapeutic area that those considerations are met. We know that in some diseases, patients very much have a community. So we want to keep that in mind and enhance the presence of these types of studies and making sure the potential studies are patient groups and communities are aware. Next slide. And again, just an extension of ensuring the presence of these types of studies and CNS studies as we apply those modern day applications and using technology to bridge any gaps, enhance education, and ensure we consider the participant, their families, and any caregivers in that approach. Next slide. So just to wrap up key success factors for operations, understanding the site matrix, standardizing kits, materials, procedures, providing ample study references that are flexible enough to fit in any institutional processes, collaborative continuous feedback, understanding site and caregiver engagement, and then also all of the above to protect our endpoints and have positive participant outcomes. And so, James, I'll hand it over to you for final takeaways.
Okay. All right, 2 messages then. So the first is that if you're at a company or an academic and you're working in these sort of vectors or ASOs, or, the trials can be done, and there are experienced investigators and folks out here to help you. The environment is favorable. We have success. We want more success. And the other one is that if you're more target oriented and you're working on trying to make small molecules that get in and hit a target, widen your perspective. Think about all these other potential ways of directly targeting parts of the CNS and the targets in the CNS that you want in order to achieve your therapeutic goals for the patients. And I think we're entering a new age here for CNS medicine that I hope will improve people's lives. So thank you all for attending and your attention.
Well, thank you very much for that very insightful presentation. I hope everybody enjoyed it. We don't have time for the Q&A right now, but I would just like to bring to everyone's attention, if you have any further questions and the questions that you have sent in, remember that you can send your information to info@medpace.com, and your questions that you sent in today, the team at Medpace will follow up with you after this presentation. So thank you everyone for participating. First of all, I'd like to firstly say thank you to James, Felipe, and Kelsey for that very insightful presentation. I hope everyone enjoyed it. Now I'm going to be sending you in your chat box there. Or actually first, I'd like to say that you will be receiving the follow-up email from X-Talks with access to the recorded archive for this event. A survey window will be popping up on your screen. Your participation is appreciated as it will help us to improve on our further webinars. Now, I'm about to send you a link in your chat box. You'll be able to view the recording of this event at that link and also share this link with your colleagues once they register for the recording as well. So I encourage you to do that. Now, please join us again in thanking our speakers, James, Felipe, and Kelsey, for that very insightful presentation. So thank you very much. We hope you found this webinar informative. It has been my pleasure to be your webinar moderator. On behalf of the team here at X-Talks, we thank you for joining us. I'm Sonia Hunt. Until next time, please take care and bye for now.